Okay, hi, welcome to Elixirgen Scientific's first webinar. My name is Mohamed and I will be moderating today. I'm a scientist at Elixirgen. Today we're going to be talking about human iPSC-derived neuromuscular research and transcription factor-based fast iPS differentiation. We're going to be hearing from two speakers, Dr. Mohamed Farah and Dr. Muzammil Ahmad. And we are so excited and thank you for joining us here today. So as mentioned, we're going to be hearing from two speakers. If you have any questions during the talks, you can insert them at any point into the Q&A section by pushing the Q&A button on Zoom. The questions will be addressed after each speaker, and it will be helpful if you could include the slide number, which are located on the bottom right of the slide, so we know what your question pertains to if it's about something specific. If we do not get to answering your question due to time constraints, we will be contacting you via email. So if your screen name is similar to the name that you registered with, we will be able to find you and get back to you. So our first speaker is Dr. Muzammil Ahmad, who is a scientist here at Elixirgen Scientific. He has a Ph.D. in biotechnology from the National Center for Cell Science in India and completed a postdoc at the NIH National Institute of Aging. He's been with Elixirgen Scientific since 2016, where he is a leader in our research and development team. And he specializes in creating easy to follow intuitive protocols, and particularly is our resident expert at GABAergic neurons, dopaminergic neurons, hindbrain, vascular endothelial cells.
Thank you so much, Mihal, for the wonderful introduction, and thank you all of the attendees, and welcome for to the first webinar of our webinar series. So today I will talk about in this webinar about our company, our technology, our products, and their characterization. So first of all, let me give you a brief introduction of our company, Elixirgen Scientific. And Elixirgen Scientific is the leader and the expert company in tissue differentiation from human induced pluripotent stem cells and stem cells. So the company was founded in October 2016, and our head office is located in Baltimore. Our research R&D team works here. Our branches are in Japan, and we call our technology quick tissue technology, because we use transcription factor for very rapid and efficient differentiation of iPS cells into various subcell types. So Elixirgen Scientific offers products as well as services. So in products, we have differentiation kits, which you can use for the rapid differentiation of your iPS cells into various cell types like neuron, endothelial cells, muscle differentiation, and astrocytes. We also have the differentiated cells from disease as well as from the healthy donors, and we freeze down the cells in the precursor state, and then we provide you these frozen cells for your research. So we have the access to several disease and healthy cell lines. So from California Institute of Regenerative Medicine. We also have assay-ready plates where the neurons of your choice are already plated onto a MEA multi-electrode array plate, and which you can use for your research purposes in your laboratory. In the services, we have services for the differentiation where you can give us and send us your iPS cells, or you can choose any iPS cell from our cell bank we have, or we can also suggest you that from where you can get the iPS cells of your choice. So it could be the healthy cell line or disease cell lines or a genome edited cell line, and we will differentiate into your desired cell type and we can send it to you. We also do the assay services where we can do the assays for you if you want to test your compound or the drug on certain neurons or other cell types. We can do multi-electrode array or ICC based assays for you. We also help in doing the R&D services. So where actually if you want to differentiate your iPS cells into your required cell types, but you don't know how to differentiate them, we can help you to differentiate your required cell types faster and very efficiently. So here the question arises that why someone should use Elixirgen Scientific quick tissue products and services. So we have not one, but multiple reasons where you will find why you should use the quick tissue products. So one is that we spend a lot of time in the systematic development of our products. We spend time so that we can make a very rapid product where a very efficient kit which can rapidly differentiate your iPS cells into your required cell types. Though this is our products are very rapid but robust also. I mean to say that we test our kits on several iPS cell lines, and indeed we found that there is no variations iPS to iPS lines what we have tested so far. Although our kits are rapid, robust, but we don't compromise on functionality of our products. So we test and we compare the functionality of our differentiated cells with the human primary cells, and then we only release our product. And with that, there is no chance of genetic footprinting. I mean to say that we use mRNA, synthetic mRNA or virus technology to introduce the gene into the cell for which we are going to differentiate. So there is no chance of the integration of that gene into the genome of the cell. And we always try to produce a very simple way to follow the protocol, so that our purpose is that even someone who is not very well trained and working with a trained person, so that a new person can also follow this instructions and do the differentiation. And we try to write these protocols in a very similar fashion, so that if someone is using multiple products from our website, so the protocol looks pretty similar, and then they should not be confused in using that. On the other hand, if by using other media-based or the small molecule-based methods or other transgene method, you may not find all these qualities in one protocol. So here I will show you how do we develop our quick tissue products. So here is the flow chart of how do we do that. So first of all, we select a cell type which we want to differentiate into a particular cell types, and then we identify the most prominent transcription factors or the genes to introduce into the cells for the post differentiation. Then we transfect them, assess the differentiation, which is followed by the media condition optimization based on the available literature. Then we validate our differentiated cell types by several methods, and after doing that, we do the functional assays for all the differentiated cells. And in the end, after characterizing our product, we do the fine-tuning of our protocol to make it easy to follow by the users. So this slide is the heart and the soul of our technology, basically. So here you are seeing a gene expression correlation matrix, and this study was done by our founder of this company, Dr. Minoru Ko, in his laboratory at Keio University. So there, scientists identified 714 genes which, after induction, can force the ES cells into a differentiated particular type of the cells. So these 714 genes were cloned into stem cells, and then the transcriptome data after the induction was correlated with the publicly available data, which you are seeing on the y-axis, and the x-axis are the 714 genes cloned in stem cells. So we found, they found actually very high correlation of these genes, particular with the tissue type. Like in this corner, you are seeing that the genes which are mostly transcription factor in this correlation matrix, the genes in this area are mostly correlated with the gene brain tissue. In this area, lung, and the down is the blood. So this is where, and Elixirgen Scientific has the license from KU University exclusively to use this gene matrix for this commercial purpose. So for example, like on the right hand side, you can see the neuron and the endothelial cells. So if you wanted to differentiate neurons, so we select our transcription factor and the genes from this region. And if we want to differentiate a subcell type, which is from the blood lineage, we choose the transcription factor or genes from this region. So here I'm going to take you through the whole process of our differentiation and optimization. So this is the example of the GABAergic neurons which I'm showing, and the same technology or same process we use for all the differentiated cell types. So here you can see these treatment are the transfections of the synthetic mRNA, and other technology what we use is the Sendai virus. The gene is cloned in the Sendai virus, which is controlled by temperature. So that also we use. So here in this process, we try to, after selecting these potential transcription factor or genes, we try to optimize the number of transfections. So in this GABAergic neuron differentiation, you are seeing that 4 transfections we do. But like for endothelial cells, we optimize 3 transfections are good enough to reduce the expression of that genes. We do the media optimization and the conditions like which day the cell should be passaged onto the cover slip or onto the plastic well. So here, this is, you are seeing day 1 picture. So where we describe about what the cell density at the start you required for doing the differentiation. Day 4, where you can see the cells are already started differentiating, and there you can see the neurites are coming out from the cell body, and that time it was passaged onto the cover slip, and by day 10 you see a fully differentiated neurons, which is followed by basically for the identification of the markers which are considered to be the hallmark for that cell type. So here we validate the cell types by doing immunocytochemistry or RT-qPCR, QRT-PCR, and we do the global analysis also of the gene expression and compare with the primary cells. So microarray and RNA-seq we use for that purpose, and sometimes we use FACS also to characterize our cells. So here you are. Seeing the this data or the RT-qPCR, where we show that with the induction of the parvalbumin and the GAD1 gene in the GABA, which are considered to be the markers for the typical GABAergic neurons. So this is merge image tubulin beta 3 expression, which is beautiful at day 10. This is the expression you are seeing, and the parvalbumin which are located into the nucleus. After characterizing all the characterization, so we try to fine-tune our protocol in a way that users who are using this, these kits or our product, we try to find a way so that users should not come. They don't need to come on the weekends, especially. But if we cannot really skip that thing, so we try in a manner that if there is a need to come on weekend, it should not be more than a media change. So that is basically just a 10 or 15 minutes a job. Your colleague, if someone is coming to the lab, that person can also perform that job. So here is the series of our product, which our team of scientists have generated after doing a lot of work. So we have a series of neurons like excitatory, GABAergic, dopaminergic, cholinergic, sensory, and sensory neurons. So by sensory, I just realized that yesterday the Nobel Prize in the field of medicine has gone to the for the discovery of pain and heat that receptor. So TRPV1, and our sensory neurons are TRPV1 positive. And recently, we also launched astrocyte, which is an amazing product because you can differentiate your astrocytes from iPS cells in just 28 days. And we have a skeletal, quick muscle, skeletal, and endothelial cells kit, as well as frozen cells. So now next, I'm going to show you some examples where we and our collaborator and our users have used our product to for the functional studies, for disease modeling. And if you want to know more about the functional aspects of our product, there is another webinar on November 4th, which Dr. Kara and Dr. Mihal will present in front of you. So please scan the barcode in the end of the presentation, and you can know more about the functional aspects of our products. So here, the slide I'm showing you is a very recently published application note on our website, which you can read on our website in detail. So in this experiment, excitatory neurons were actually transducted with GFP lentivirus, and GFP was cloned under a brain-specific promoter. So here you can see the expression of the expression of GFP intensity is correlated with the MOI of lentivirus. So higher the MOI, higher is the GFP intensity in the neurons. So there, by using this method, this invasive method, you can tag your protein with GFP under this promoter, and then you can use for your analysis this kind of analysis. So here, this data of PESCLEM to study show the function of the single neuron was provided us by E5 Science, where they showed that this the single neuron has the firing activity, and the post synaptic current was observed in the single neuron of single excitatory neurons at 5 to 6 week differentiation. So this data is courtesy from our collaborator, who's Rico, and in this experiment, they used the neurons already plated on this MEA plate, MEA48, and they plated we plated excitatory neurons actually. And this is, if you know want to know more about this experiment, please, again, I would like to say that please join us on November 4th, Thursday, and then you can find more details about this data. So here they tested agonist and antagonist of neurotransmitter, and then look for the relative number of spikes, and the excitatory neurons, they responded to these agonist and antagonist as it was expected in a dose-dependent manner. And here, this experiment was done in-house, and this is the differentiation of a healthy line. Cholinergic neurons were differentiated from a healthy line, and ALS IPS from an ALS patient line. So this is SK011, and after doing this, and then we did the ICC for the localization of TDP43, which is considered to be the pathological protein in case of ALS. So here you can see the lot of aggregation of TDP43 in the cytoplasm of the soma. Another example again is from the ALS disease line, and this work was done in collaboration of Johns Hopkins University, where this work was published in Frontier of Cellular Neuroscience. There, the scientists, they used iPS line from a patient, a gene-edited line mutant, and a healthy donor lines, and differentiated cholinergic neurons, and co-cultured these cholinergic neurons with human primary astrocytes in a MEA plate, and then they the firing of action potential was recorded for 15 minutes, and they found that the mutants were, they showed lesser number of spikes and diminished network bursting compared to the healthy donor lines, the different neurons. So this is how people have used our product for disease modeling. Another piece of the data I'm showing you is again courtesy from our collaborator Rico, and where they differentiated a healthy cell line and Alzheimer's disease patient cell line into excitatory neurons, and they measured the release of A beta 42 into the supernatant and the tau protein into the cell lysate, in cell lysate, and they used ELISA for measuring the amount of these two peptides and the protein. And indeed, they found that the AD neurons differentiated from the AD lines have more of, they showed the more release of A beta 42, and the cell lysate contain more amount of tau. Last but not the least, this study was published in Molecular Therapy, and here the users, they disease model for myotonic dystrophia. So they differentiated a patient IPS line using a quick tissue muscle kit from Elixirgen Scientific, and differentiated patient line and a corrected line using the same kit. So the phenotype for disease for this disease is the RNA foci because of the CTG repeat expansion in the protein kinase gene of this disease at the 3' UTR. So you can see the RNA foci in the differentiated muscle in the nucleus, although it is disappeared in gene corrected lines. So by the beauty of this is this data is that they got this data really very quick by using the only after day 7. So this is the picture at day 7, which might have taken a longer time to see this kind of data. So here, the take-home message is that that Elixirgen Scientific quick tissue technology indeed is very robust, rapid, and functional. So here after that, I would like to take question and answer from you, and I hope you enjoyed the presentation. And again, I would like to welcome you for the next webinar series, and then get ready for the next speaker too.
Thank you so much, Dr. Ahmad. So remember, please ask any questions in the Q&A box, and we will address them. All right, so here, Dr. Ahmad, you mentioned that Elixirgen has both SEV and mRNA kits. When would someone choose one over the other? Was there an advantage of one?
So yes, essentially they both are same. So we use two methods for the gene introduction. So one is synthetic mRNA, another is the gene is cloned in Sendai virus, which is controlled by the temperature. So I would say that Sendai virus kit is there is a little less work because it's a one-time infection, and then users they are free to go, and then after that it's just a matter of the media change. Whereas in the mRNA-based kit, users they have to do 4 transfection during the time of 2 days. But as per the requirement, people choose mRNA as well as Sendai virus kit, because some of the labs they really don't like to use viruses, though the Sendai virus is perfectly safe because the human is not the natural host for Sendai virus, and rodents are. So people who are working on rodents, they really are careful about that. Otherwise, they both are very efficient and robust.
Thank you. How long are you typically able to keep differentiated cells in culture?
So, so far, in one of the experiment I showed here in the functional disease, the data, so ALS and the human astrocyte co-culture, the Johns Hopkins data, they use for 6 weeks, and we have even used longer than that if just perfectly fine neurons onto the cover slip.
Okay, let me see questions. Oh, here, if someone has a particular gene mutation that they're interested in, or disease model that they're interested in, where can they obtain the iPSC lines for themselves to differentiate with a kit, or to send to you to differentiate?
So basically that we can answer you if you want that kind of help. We have sources and we have information which we can suggest you if you want a particular mutation or mutated cell lines where you can get it. So I would request you to email us on our you can find on our website on maybe probably in the end of the presentation. So please email us. We will provide you that detail if you are interested in particular IPS lines. And then, yes, of course, you can purchase the kits from us or we can differentiate those cells for you.
All right, I think we have time for one or two more questions. What surface protein coatings do you use? And is the cell medium fully defined? Does it contain serum?
So the medium is serum free and we don't use serum containing medium, and we use coating material the PLO and laminin for neurons, and we use we call it coating material A, and which is that coating material is also available on our website, which is called iMatrix Silk. We use for IPS coating, and for neurons, I already told you we use PLO, that thing, ornithine, polyl ornithine, and laminin for neuron adhesion. Yeah.
Okay, and typically how many differentiated neurons do you get using your kit?
So so that's a good question. Actually, how many neurons? So usually from it depends which kit you are using, virus kit or the mRNA kit. So using mRNA kit, you get more number of neurons, but there are some other kind of cells may come, but that is taken care of by the media. On the other hand, which is around, I would say, one kit gives you 500,000 cells after differentiation, and so is true with the Sendai virus kit. So I would say a 4-well kit, if you are using, you get around 500,000 differentiated neurons or cells.
All right. All right. I see there's another question about MTA agreements from CERM. So we will get back to you, or you can contact us. But we pay a royalty to CERM and have access to their lines. Users do not need to pay that. All right. Thank you so much, Dr. Ahmad. Next, we're going to be hearing from Dr. Farah. So Dr. Farah is an associate professor of neurology at Johns Hopkins University School of Medicine. He has a Ph.D. in neuroscience from University of Michigan in Ann Arbor, completed a postdoc in the Department of Pathology at Johns Hopkins University School of Medicine, and his lab studies axonal regeneration in the peripheral nervous system. They are currently working on interactions of target tissues in human axons from iPSC-derived neurons, and he is going to speak to us today about the development of human iPSC-derived neuromuscular circuits. Thank you so much, Dr. Farah.
So thank you for inviting me to speak today. So what I'm going to do today is at first sort of line out why we're interested in human iPS-derived neuromuscular junction. Traditionally, we are a lab that use mouse models to study pathogenesis of diseases for a peripheral nerve system. So in the peripheral nerve system, what happens is that no matter what the disease underlying cause is, the first sign of degeneration happens at the distal where the neuron contacts the target tissue. So whether neuropathy is caused by the malneuron or axonal neuropathies, the initial sign of the disease is disconnection of the muscle or the skin and the neuron. So the target denervation happens first, and then axon is degenerate. Obviously, that's happens in trauma and injury. The other disease that this sort of phenomena happens is the motor neuron diseases, where early stages before the motor neuron is degenerated, there's a disconnection between the muscle and the neuron. So using mouse models, we have been trying to understand how did that come about and whether we can interfere with that process so we can prolong the time that the muscle is innervated. So I'm going to just show you a few things we did, but essentially what happens in ALS, which is a most common motor neuron disease, is that eventually the motor neurons will die. But before that, the muscle is denervated. And when there's a disconnection between muscle and the neuron, there's no communication. And that's where the disease signs initially start. So there are different mutations that are inherited and cause more ALS. Some of it is well known, and I think that the previous speaker mentioned SOD mutant that linked to ALS, and CO, which accounts for a lot of mutation also, even though the studies in mouse models hasn't clearly shown this, but muscle denervation seems to be happening in there. So as I mentioned earlier, I think that even though how does disease happen is varies from mutation in given protein than the other. What they have in common is muscle denervation and axonal degeneration. So for a while, we have been studying, along with others, with the mutation is mouse models that have SOD mutation. This is mouse model that have been around for a while and is well characterized, both of the molecular and cellular dysfunction that happens in this mouse model. One of the things we have been interested in is what happens at the target at early time point. So if you were to look at the muscle and stain for neuron here, for axon here in green, and for the muscle, the neuromuscular junction, the postsynaptic side in red, what you would see is that you see this co-localization of the axonal terminal with the postsynaptic side. Okay. And that's normally what you see throughout the life of wild type. In SOD mice, before motor neurons degenerate and show dysfunction, what happens is that some of those neuromuscular junctions are denervated. That's axons dies away from the terminal. You can see some of the axons still intact here, where you see this denervated postsynaptic sites. And eventually, the axon will fragment, and all the neuromuscular junctions or at least substantial number of them will be denervated, and that will lead to death. So one of the things we've been interested in is, can we slow down this process of neuromuscular denervation? And these are published works. I'm just telling you sort of to lay down why we got into the human iPSC-derived neuromuscular synapse that we're trying to develop. So one of the drugs we tested before was beta-secretase inhibitors, which are known from Alzheimer's, but also the base one, the protein, the enzyme that gives amyloid precursor protein, is expressed in the peripheral nerve system. And through studies we did with the knockout overexpression and base inhibitor, one of the things we discovered is that inhibition of base activity leads to better axonal regeneration. So we tested in SOD mice and trying to see if it will enhance re-innervation of the muscle at early time point. And what we found is that when we treat these mice for 1 month or 2 months, we see a lot of innervated neuromuscular junction. So this doesn't deal with the mechanistically what the SOD is doing. What's going on here is that inhibition of base one is allowing axons to regenerate and re-innervate the target. And we see similar things in 2 months. It's less effective at 2 months when motor neurons start dying. Obviously, when motor neurons die, there's no axon. And functionally, we can record from these muscles and see enhanced amplitude in the muscle. So these are the studies we already published, and what we find in here is that some of these drugs that enhance regeneration and repair can enhance the regeneration in mouse model of ALS, SOD mouse model here. However, as most of you are familiar with, one of the issues with the mouse model is that a lot of what's done in the mouse model cannot be replicated in human. So what we decided is that try to establish a neuromuscular connection, neuromuscular synapses in the context of human iPS-derived cells. Fortunately for us, we are here at Hopkins, and there's a large center of ALS center that studies, a large group within the ALS center that study iPS-derived motor neurons. So what we wanted to do is to take advantage of the resources around us and try to establish neuromuscular connections there. Okay, so this is one way that base enhances its powering, but essentially the questions we ask in there is looking at different mutations, different lines. There's about 50 different lines that are available for us here. Do we see difference in axonal regeneration at the baseline between specific mutants and their isogenic controls? And additionally, can we establish a neuromuscular synapse in a dish of motor neuron and iPS-derived muscle? And try to understand if we can recapitulate sort of and understand what's going on, what happens in humans in a dish using human cells. That's the goal. That's how we got into trying to develop this project. So, as almost all of you know, is that iPS cells can be differentiated to neurons. What happens when you differentiate them to neurons and put them in a dish or a glass cover slip is that you see the neurons clustering, and you see axons going from one cluster to another cluster. And what we wanted to do is trying to have in a way that axons are directed toward growing to certain away from the cell bodies. So we started using these microfluid chambers. The advantage of them is that you can place the neurons in a cell compartment and have channels where the axons can go through. And when axons can go through there, in the other side, you will have compartment with axons. So you kind of separating the axon from the cell bodies. And the idea is that we can then, for example, cut the axons and look at the regrowth. And when we do this experiment, what's shown here in B is experiment where the cell bodies are out of the view, but you see the channels, these channels where the axons are coming through. These are human-derived iPS neurons that we got from ALS center here. And then you see axons growing out to the other side. If we injure the axons here, the axons die back a little bit, but they're present in the channels. And immediately after injury, you don't have any axons growing in here. But what we can do is we can follow axonal regeneration as they come out from the channels, both live, like I've shown you in this video here. And we can also stain and do some staining that show you specific markers for this axon. Another thing I want to tell you is that since we have pure axons here, we can do sort of a molecular profiling of regenerated axons or the axons as they naturally grow. So when we follow this axon, we can understand the dynamic of growth, the rate of outgrowth. Is there a difference between given mutation and their associated controls? And do sort of analysis to them and look at certain hours. So now you see this at 0 hour, there's no axon grown out. 5 hours, you see more, 10 a little bit more longer, and so forth. And then we can analyze the average rate overall, and we can follow individual axons and the dynamic of that axon growing throughout the time that we do the live imaging. And obviously, we can do some staining and look at markers that axons expressed there. But the other side we're interested in is that do these axons actually contact muscle? If you culture muscle fibers with them. So initially, when we did this experiment, we planted muscles sort of close to axons. And what we see is that axons can grow over the muscle. They seem to be making some contact. And other beavers have done similar experiment where you can get contact and you can get transmission. But what we're interested in is, can we develop a robust system in a way that we have efficient differentiation of neuron, efficient differentiation of the muscle, and also take advantage of the knowledge that we will gain from developmental neurobiology, that's molecular that are involved in muscle formation. If we put back in this system, do we get more robust neuromuscular junction? So to that end, we want two different systems, little bit variation of the microfluid device system that we've been using. And what we did here is that we just have a longer area that axons can grow through. And we put sort of separation where we have proximal axons versus distal axons. And this is, we think, will be helpful in terms of when we do molecular profiling from what's going on at the terminal versus axonal part or neuronal part. So anyway, so right now what we add is we can get these neurons grow very long axons in these chambers, about 10 millimeter or so. And then so we turn into Elixirgen. It's because what we're interested in is that, as some of you may already know, is that when you differentiate these cells, not all of them become neurons. And additionally, what we want to have is that we want to put differentiation of these neurons so they can put out axons quickly that can get very long to reach a muscle. So to that end, we turn into our colleagues here in Elixirgen, and we tested their kits. And when we use the messenger RNA-based kit, what we're finding is that we're finding the robust differentiation of CHAD positive, which is a surrogate for motor neuron marker, as shown here. And we can see the CHAD positive axons growing out from this cluster of neurons. When we put this, so these are IBS cells that we have from our center, and we use the kit from Elixirgen. When we put them in a device, and you sort of see them here, because this is a cell compartment, this is that is nuclear staining here in cell compartment, and we stain them for islet, another marker for motor neurons. What we found is that about 75 to 80% of these clusters differentiate to islet by islet 1-2 positive neurons. So right now, what we're getting is efficient way of, and a quick way of differentiating our lines we have toward motor neuron light cells. So, and then, so we turn into the muscle, and we also use messenger RNA kit for skeletal muscle. So these are IBS cells. After 7 days of differentiating, we can get muscle fibers. And when we do staining for markers specific for muscle fibers, we can get robust differentiation of the muscle. So you can see these multinucleated muscles that are positive for markers for mature skeletal muscles. So right now we have both sides of the equation. We have efficient way of making muscle and efficient way of differentiating motor neurons. What we're doing now is that using different mutations, we're trying to put the muscle together with the motor neurons and culture for longer time and see if we can find neuromuscular junction. But also what we done to these cells is we try to pre-pattern. So normally during development, what happens is a muscle before the axon is interfere, it becomes pre-patterned by forming cluster of acetylcholine receptor. So we added to our muscle cultures some of the molecules that are known to cluster acetylcholine, and we find these clusters of acetylcholine that are present in the muscle. And right now, the ongoing experiment of putting the motor neuron with differentiated through our Elixirgen kits with the muscle of the same mutation or a control muscle, and trying to figure out what's the innervation ratio in there. Is there any difference? Using microfluid devices that we can grow these long axons. And another experiment we're testing is that the system also results in functional neuromuscular junctions by depolarizing the motor neuron and recording the calcium influx into the muscle. And so these are ongoing experiments that the May graduate student is doing now. But what we got so far is efficient way of differentiating different iPS cell lines. So we are in the go-ahead stage of the experiment. So most of the work I shared with you today was done by Madison James, who's a graduate student in my lab. Catherine Marshall started this project, and some of the mice works were done with previous grad student in the lab, Carrelation. The devices we use, they're locally made here at Hopkins, and they're made in Aaron's lab. The cell lines we get it from Merkakis and Ratstein lab, which are in the same floor with us. And thank you for listening, and I'll be happy to answer any questions that you may have.
Great. Thank you so much, Dr. Farah. So again, people can ask their questions in the Q&A. We already have some questions that came through. So which media is used in the different regions in your microfluidic chamber? Is it the same media throughout or a different set?
No, so what we've been doing is that we use for the muscle part, we use the muscle media. And axons seem to be growing actually very well. I haven't shown in here, but we think that there may be some unknown factors that axons are getting from the muscle, because they seem to be growing very well across. So let me show you this experiment where I show you that they grow. This is muscle media on this side and neuron media on this side. And for some reason that we're trying to figure out is that we get more healthier axons growing on top of the muscle.
Is there any sort of coding? Do you use any coding in your neuromuscular junction for the motor neurons? Or
coding? Yeah, so the coding we use is actually, what we use right now is laminin. We use laminin for both.
The skeletal muscles that you use, are those iPSC-derived or primary cells?
So these are iPSC-derived, these muscles. And that's why we turn into this kit.
Right, so you use the Elixirgen Scientific kit.
Yeah.
And how long are you able to maintain the system?
So far, we can maintain it up to 3 weeks. Our hope is to do it longer.
And are there any viability issues with either the muscle or neuron differentiation?
So the major, not the neuron part, the major part we're trying to optimize now is the muscle part, because what happens is that if you put too much of this muscle, it just come off the dish. So right now we're trying different.
Differentiation itself worked well. Just once you put it into your model, something.
Yeah.
All right. Does your iPSC skeletal muscle cells do they express fetal or adult forms?
So the fetal or adult forms. So Titan is a differentiated marker for skeletal muscle. So these are what they express. Is that the question, or do I miss? I missed something about the fetal.
Okay. And do you use any alpha bungarotoxin binding to discriminate between fetal and adult forms?
So these are alpha bungarotoxin. So we do. So green here is alpha bungarotoxin staining.
Great.
So that's it, yes.
Great, thank you so much. I think we are good on time. And if anyone had any other questions, we will be emailing you or answering that way. Thank you, Dr. Farah. So if you can. So we just wanted to, again, thank you, Dr. Ahmad, Dr. Farah, for your excellent presentations today. We wanted to remind everyone that they can join us next month for another webinar that will focus more on the functional aspects of using our cells, modeling neurotoxicity and neurological disease using the functional iPSC-derived neurons from Elixirgen Scientific. We will hear from Dr. Gordon, who's the director of neurobiology and at Vala Sciences, and I will speak for Elixirgen Scientific. That webinar, as well as today's webinars, will be available on YouTube for people who missed it, or if you want to watch it again, because it was so great. If you want to register for next month's webinar, you can scan this code, and if you register within the next 24 hours, you will not have to go through any of these screening questions, which you had to for today's webinar. After 24 hours, we will be putting those screening questions back on so that we can gather data to best serve our customers. Anyway, thank you so much for joining us. And, again, please contact us to let us know how we can best serve you and help you proceed with your research.
So thank you. All right. Bye.
All right. Have a wonderful day, everyone. And next month. Bye-bye.